So I will also, so I have also done the recording for the session and okay, so I was expecting much less people to turn out because of the very interesting match that has been scheduled for today. So I was hoping like a lesser audience only. But anyway, we will have to go on with our discussion. So we'll have a regular session. And okay, so let me start. So by now, I think the like how I conduct the sessions that is fairly similar, right? So first of all, what I'm going to do is okay, sorry. So first, I'm going to share my screen, and we will actually discuss the presentation for. Okay, we will discuss this particular presentation. Yeah. Okay, so since people are still joining in, let me just stop sharing for a while. So meanwhile, let us discuss what we will do today. First of all, we will solve the previous year's question for the week 3 content. So these are the questions that were given as an assignment last year. To people who are new, we do not generally discuss the current assignments because usually the session is taken before the submission date for your assignment, right? So it will be an unfair grading if I discuss the same questions. But a lot of times what happens is that some questions are recurring and more often than that, what happens is the concept on which the question is based is the same. So if you understand the concept of how that question has to be dealt with, I believe you will be able to better understand your current assignment also, right? Apart from that, there are some extra topics that we talk about. So these topics include some other maybe concepts that were touched upon in the course slides or in the course videos, but they were not talked upon in detail. So I thought I'll introduce those concepts in a more comprehensive way over here. Or they are some of the doubts that remain. For example, the kind of doubts that people raise on the discussion forum, right? Or sometimes what we are also doing is that if in the previous class somebody has some concept that has not been cleared out, I have tried to incorporate those doubts into the next session so that we can actually clear out those doubts as well. Um, yeah.
Madam,
So, yeah,
just today, maybe some loose talk. Somewhere I
yeah.
saw China
Yeah.
has come out with a better than CRISPR technology. So next
Okay.
session when you are taking, if at all you take,
Yeah.
kindly describe what is different, what
Mhm.
are the advantages and things like that. I know it may not be
To,
useful for this course, but it will be
yeah.
of interest, general interest to know that.
If we are talking about science, it is always going to be useful in some way or the other. But could you like give me a name for what you are talking about?
Today I saw in a WhatsApp message saying that Chinese people have published in Nature a better
Okay.
technology than the CRISPR, which
Okay.
is going to revolutionize this field. That's what I
Okay.
just read, but I did not
Okay.
go detail into that. You are
No, it's
the
fine.
best to do that. So maybe
No.
one slide you can prepare next week, next
Sure.
session,
So,
and share
yeah.
as it may not be useful for this course, but it is exciting to hear that newer technology is coming even at this stage. That's what I just wanted to
Yeah.
mention.
So
Thank you.
actually, there are a lot of up and coming technologies also. For example, there are some that are not covered in this course, right? So that is also due to the restriction of this time duration that sir had maybe. And along with that, also, I think if I'm not wrong, the course has been running since 2016. right? So it has been a while since 2016. So a lot of advancement has basically happened in the field of science also. For example, there are other imaging techniques also, like optogenetics that has come up, which is one of the things I think which is very, very interesting to read, but it is not a part of this course right now. But maybe if it was devised at this particular point of time, it might have been a part of this particular course. Similarly, there are obviously newer technologies coming. There are a lot of talks about omics now, right? Different kinds of omics. So now, because we are not anymore restricted to transcriptomics and genomics, right? So people have moved on to metabolomics because they are giving a more real-time understanding of like what is happening inside our body. They are if they are basically assessed, it is the genotype to phenotype correlation is a little simpler. So these things are there. We had actually initially discussed with NPTEL in one of the session with the PMRFTAs that we might take an additional 1 week. That I am still not sure about. That if apart from these 4 week courses, if we are taking that additional 1 week, but if we are, I plan to inculcate these things in that particular session. But yeah, so what you have just told me about, I think I'll go back and I look at Nature News and Views. If I can find that, I'll definitely include it.
The other additional point, I went through the
Yeah.
syllabus, week 4 material is available. I saw that for a molecular biologist or a biochemist, these are all perfect. 4 weeks, you have
Yeah.
squeezed so much of material. But my feeling is for the general public who, like me, I'm a computer scientist and trying to learn something in this field, it should be made into 8 weeks course, not this 4 weeks, because too much
Yeah.
is said and then suddenly so many technology exposed. By the time we understand what is nucleoside, nucleotide,
Yeah.
everything goes above our head. So best thing is to expand this course to 8 weeks, includes
Yeah.
little bit more material, but definitely some exercises and other thing should help us. But the course is very fascinating. The second week assignment I did and fared,
Oh,
very low, I should say. So even
okay.
though I made all guesses, this and that, and then little
Yeah.
bit of chat GPT, etc., I scored very
Okay.
low. So I thought that
Okay.
I should give a feedback to all of you, saying that it should be made into a 8 weeks course.
Yeah.
Little bit, because so much of material squeezed
Yeah.
into these 4 weeks. Of course,
Yes.
it is
Yes.
not decision is not mine or yours.
Yeah. It.
Definitely, you can give a
Uh,
feedback to your professor, and
mm.
you people are very good, professor as well
So
as all the
I am
teaching
not really
assistants.
sure if
But
it is
how
up to
to?
sir as well. Like I am not sure how it was first devised. There are a few courses that I know about, and the ones that I have come across, usually the 12-week courses are the ones that are courses like basics in biology or basics or introduction to a particular field. These have been given more time, and I think more specialized courses are running for 4 or 8 weeks. Now, how that was devised in the beginning, that I'm not really sure about. But yes, if I can, I will pitch in this to NPTEL itself.
So I leave it to you to take over and then continue the
Yeah.
session, madam.
Yes, yes, sure. Okay. So, yeah. So we will first begin with our first presentation, wherein we will basically discover the previous week year's assignment, which was basically devised for this week 3 content, right? So let us go through this assignment, and there are some techniques that are being talked about over here that have been talked about in the course as well. So wherever we are going to encounter any technique or any term that could be ambiguous for us, we are going to stop and discuss that also, right? Okay. The first question for week 3's previous year assignment was, shown here is a schematic of the image for a DNA sequencing gel using the Sanger method. Based on your understanding of the methodology, identify the option that shows the DNA sequence of the template strand. Okay, and you have four options that have been given. So, since this is something that you will require a little bit of time to solve, I will maybe remain silent for a minute. Try to solve this. I would like to give you two hints while you are solving. So one, you have to be very careful about the 5' and the 3' end, right? So obviously you will have to have a little basic understanding of where in this gel you have to look for 5' end, wherein you have to look for 3' end. Also, another hint from my end is that you have to read the question very carefully, right? So what has been asked in the question statement? They have asked for which kind of a strand, like, okay, so I will not go much into the detail. I will give you guys a little time to solve. Yeah.
Madam, while solving this type of problem, I
Yes.
found that just the first four, you know, 4 letter or 4 sequence is sufficient to give the answer, not necessary to go through the whole sequence, looks like.
Yes,
Of
yes.
course.
So over here,
So,
yes, that is the case, because over here we are not really getting into competitive exams. In competitive exams, sometimes what they do is just to confuse people, they do have similar sequences or similar these dinucleotides in the same places. But over here, if you get the first 4 or 5 correct, you will be able to do it.
so I
Right.
could solve this one as answer is B, GACG, something like that, because I can
Yeah,
easily
yeah, yeah.
see from top. C is the first, CTP is there. So C opposite that is G. Then next level is TTP is there. T means next A has to come.
Yeah.
Then GTP is there. So again C will come. So like that, if I just
Yes.
see GAC, G is only
Mm.
one type. I did not check the whole other system. So
Yeah. So
bluntly,
in
B
this,
can be
you
the
do.
answer.
Yeah, so B is actually the answer, and actually you do not have to go to the
Whole
options that
sequence.
are given in these kind of problems, right? So over here, the how the problem is generally solved is by this method only. So you have basically a gel. You will start listing out what is going to happen in that gel, right? For example, just for people for whom this question might not have been clear, over here, what they have asked us to do is, okay, just a moment. Yeah. So what they have asked us to do is look for the template strand. Okay. So template strand meaning what? Meaning the strand that will be complementary to the one that has been run in the gel. Right. So in the gel, let us first see the sequence that has been run in the gel itself. So over here, you can see, if you zoom in, you can see the first base is C. Okay, I am unable to annotate. Uh, Sorry for this. Yeah. Okay. So the first base over here, you can see that it is C. The following one is T. After that, it is G. And the following three bases are C, T, and C again, right? So this is what this particular DNA strand sequences that has been run on the gel. On this gel, this positive end will be the 5' end and the negative end will be the 3' end, right? Now, do we have to jot this particular answer down? No, we have to basically look for the template strand, right? So what will be the template strand? It will be a strand that is complementary to the one that we will obtain in this particular sequence, right? So in this complementary strand, since we know that DNA is reverse complementary, so 5' end will lie on the negative side and 3' will lie over here. And we can, through a simple complementarity, we can basically say that the sequence should be G, A, C, G, A, and G again, right? So this one satisfies, this option satisfies this particular uh, uh, sequence, right? So that is why we know that the answer that we have been looking for is B, right? So it was very correctly solved by Balaji, right? I hope the others were also able to solve it, or that at least it helped the others, right? So over here, the sequence of the template strand should be reverse complementary to the sequence that has been derived from the reaction. So that is the first thing that we learn. Also over here, I have basically made a slide so that when you receive the PDF for this particular question, this is very apparent or clear to you how this answer was actually obtained, right? Okay, so coming on to question number 2, which basically says that in chromatin immunoprecipitation assay, that is the CHIP assay, the protein DNA complex is pulled primarily using which one of these? So is it antibody? Is it glass beads? Is it special affinity column? Or is it a cross-linker? So anybody who has actually gone through the CHIP assay might know that these are all terms that one might encounter in this particular technique, right? So all of these things are used somewhere or the other. But over here, a specific function has been asked, like we have to assign a specific function to one of these things. The function is to pull down the protein DNA complex. So which one will be the answer? Okay, so let me actually come to the answer. It is going to be the antibody, right? So first, let me read out this particular definition of chip that I have picked up from CST, which is one of the companies which provides basically. Okay, there's somebody trying to join in. Wait. Yeah. Okay. So I'll share my screen again. Yeah, we were here. Yes. So, yeah, this but I am unable to. Okay. Do you guys get a notification at around this point? Because there was several people who joined. Okay, same person. Okay, sorry for the interruption to the rest of the learners, right? Okay. So we were coming down to the definition of chromatin immunoprecipitation. So chromatin immunoprecipitation or CHIP is basically an antibody-based technology which is used to selectively enrich specific DNA binding proteins along with their DNA targets, right? So what happens in this particular case is I have basically given a very pasted a very rough flow chart of what happens in CHIP so that we can understand in which step basically the pull down actually occurs and where this antibody is actually used. Okay. So in an in a broader overview, what happens in a chromatin immunoprecipitation is first step that occurs is cross-linking. Now, what does cross-linking mean? So cross-linking basically means that we have to basically fix the proteins that are basically interacting with the DNA in that position itself, right? So for that, we will perform some kind of fixing so that that protein is which is already interacting with the DNA is actually present there as a unit itself. So this particular phase is called as cross-linking. Thereafter, we basically shear this long chromatin process. Okay, so we fragment them. So this fragmentation can either be by enzymatic digestion, or it could be through a technique such as sonication, wherein a large basically chromatin will be broken down into smaller fragments, right? After this, after we have obtained fragments of chromatin, right, which are basically interacting with certain proteins. After this, we will perform the immunoprecipitation. So since this particular, if we break down this word, it says immuno, right? Immuno has something to do with antibodies. So that is how you can remember this. Okay. So, Balaji, do you want to say something?
I just wanted to know whether this antibody is interacting with only DNA or with protein DNA complex.
No. So the antibody will be interacting with the protein actually.
In that diagram that you have shown,
or it could be the protein.
In the diagram that you have shown, that red diagram, immunoprecipitation, so-called antibody is only interacting with the DNA chain.
Okay, so this is a very brief workflow. Most of the antibodies are actually targeted to proteins. Okay, and sometimes they can also interact with complexes.
Mm-hmm.
So
Anyway,
over here, for example,
Anyway, I just wanted to know because illustration said only DNA. So I was thinking that I should next time they flip the coin and then say whether it's interacting with protein or interacting with DNA, then what to do. So now I understand. So it must be complex probably for both DNA and maybe protein, maybe both. We don't know. Okay. Thank you.
Yes. Okay. Okay. So, what I was explaining was that this particular definition, this particular term, that is immunoprecipitation, if we divide it, the first part says immuno. So, immuno has something to do with antigens and antibodies, right? If we go back and recall immunology as one of the subjects, right? So, it has something to do with antigens and antibodies. So, from here, we know that we can remember in such a way that this particular step says immunoprecipitation means precipitation of some kind of a substance using antibodies, right? So that is how we can remember it. And precipitation is essentially to pull something out of one phase and basically, for example, if we have a liquid and we are precipitating out certain solutes from this liquid. So that would basically mean extracting out or pulling out those particular solutes from this particular solution, right? So over here, since this precipitation essentially means taking away or taking out these protein DNA complexes with which these antibodies are interacting, this particular step is also known as pulling down, right? After this, whatever component has been precipitated or immunoprecipitated will be purified. It will be washed first so as to remove the excess material that has not basically bound with the antibody. Then it will be washed, and these glass beads using which it had been initially pulled down, they will also be removed, and a purified DNA will be obtained. Now, with this purified DNA, what we can do is we can basically either just see we can apply a PCR for this particular DNA, and we can basically visualize what kind of an expression it is giving, right? Or we can send this DNA for sequencing, so as to basically understand the kind of domains, like domains that have not previously been discovered, but we want to understand that what kind of domains this particular DNA segment has, such that it could harbor a protein, right? So for these things, this chip analysis could analysis could be done, and the chip assay could be useful. So this is a simpler but more detailed explanation, wherein they have explained that there could cross-linking is the first step that occurs in chromatin immunoprecipitation, after which there is lysis or shearing of the chromatin. Thereafter, there is antibody binding. After the binding of antibody, immunoprecipitation occurs, after which this immunoprecipitates are washed for a couple of cycles, so as to remove this unwanted fragments. And this after that, the crosslinks are reversed, the proteins and DNA are unbound, and DNA are purified, and then they could lead on to the analysis for this particular DNA and protein, right? So coming on to a third question, which is, which one of the following option correctly identifies the potential of the popular bioinformatic tool, the multiple sequence alignment, or MSA?
Excuse me,
So is
madam.
it, yeah,
The previous question,
yeah,
where glass beads, where special affinity column, where cross-linkers are used, is there some other, in the question number 2 that you were discussing?
yeah.
Glass bead, special affinity column, cross-linker. Special affinity column maybe for peptides. I don't know what is this cross-linker
Yeah.
doing.
So the
And where,
cross-linker is actually in the first step, it is used for fixing the proteins to the DNA, to the chromatin. It is being
okay.
used in the first step itself, right? The
Uh-huh.
glass beads are being used to purify the DNA, but wherein they are added, which step particularly, I will not point out right now because I am not very clear with that, right? So
Sure,
I
sure.
don't
No
want to
problem.
say
Just
anything indirectly.
out of
Yeah,
curiosity, I asked where they can
yeah.
topple us. Asking
Yes.
them. Thank you.
As for the special affinity column, it is one of the terms that has been picked from DNA sequencing, right? So as to confuse, create a little confusion over here. But crosslinker and antibody and glass beads are definitely a part of the chip assay. Crosslinkers are used in the very first step, wherein they are fixing a particular protein to a chromatin, and antibodies used in this particular step, which is
Okay.
immunoprecipitation.
Thanks.
Glass beads, I will verify this and I'll get back. Yeah. Okay. For the third question, which one of the following option correctly identifies the potential of the popular bioinformatic tool, which is the multiple sequence alignment, or the MSE? So this was one of the tools that was discussed in the tutorial session, right? So let us go through the options once. The options are: it can identify the sites for creating genetically modified model organisms, or it can help to identify amplified regions in the genome, or it can infer homology and evolutionary relationships between the aligned sequences, or all of these.
Looks like
I think this,
C is the
yeah,
answer. C seems to be the
yeah,
answer. But I wanted to
yeah.
ask you regarding
Yes,
this. When you are saying
mm,
evolutionary relationship, is it very similar to phylogenetic trees or different or
yes,
same?
yes. So,
Okay. I should not be asking that
this is
question, but I
actually,
am asking that.
no. So actually, it is a very valid question, which is why I have actually subsequently, along in continuation with this particular question, I've also introduced phylogenetic trees. So phylogenetic trees are, yes, they are evolutionary trees. They are one kind of a representation of how basically evolution has occurred. And the basic like material to create that the phylogenetic tree, it comes from multiple sequence alignment. So how much anything aligns, like among multiple species, how much homology is there between sequences? That is something that is given to us by multiple sequence alignment, right? And after that, how we use this sequence is by basically creating these evolutionary relationships, which is basically phylogenetic trees. So that we'll discuss about in the upcoming slides. So yes, the correct answer to this indeed was that it can infer homology and evolutionary relationships between aligned sequences, right? Is there somebody in the lobby? Like I just received a notification. So I will share again. Yeah. So we were on this question and we had basically found out the answer for this particular question. But in relation to this, I would like to discuss a little more about multiple sequence alignment and about phylogenetics. So multiple sequence alignment is basically a tool that is used to identify evolutionary relationships and common patterns between the genes. Along with this, it also refers to the sequence alignment of three or more biological sequences, usually DNA, RNA, or protein. So either it can align proteins with proteins or DNAs with DNAs for multiple different sequence species, right? Also, alignments are generated and analyzed with computer algorithms, right? So, for example, this had been discussed in the TA tutorial session, but again, we will first take an example of histone 1. So, histone is histone H1. So, histone is one kind of a protein. And histone H1 is one of the proteins that is involved in basically wrapping up of the compaction of the chromatin, right? So if this particular histone H1 is looked at, which has a residues of about 120 to 180 in length, then we can see that it is mostly a very conserved protein, right? For example, over here, wherever asterisk mark is present means that it is conserved. It is a conserved amino acid across all these species, which are humans, chimps, mouse, rats, and cows, right? So across all of them, this is completely conserved. Apart from that, there are some other dots that are mentioned. For example, these two dots, which basically signify that there at two places out of these five, there are some different amino acids. But again, those amino acids are also same. Like it is not that one place has T, the other has A, and all the other three have S, right? They are also similar among one another. So it basically still is conservative. It is not as conservative as these asterisk marks, but it is still conservative. Also, there are some places wherein these residues are non-conservative. For example, humans and chimps have this particular protein residue, but all others have okay, but this is like conservative. I think they mislabeled this. So I am not. I will probably correct it in the slide.
Non-conservative
So
is okay, madam, because
yeah,
leucine is there and proline is there. Proline is a restricted amino acid. Leucine is
yeah.
like flexible, so therefore it is
Okay, they do
non-conservative.
not have a similar function also. So it is a not a missense kind of.
It is a constrained amino acid, so that's why they have
Yeah,
said
yeah, 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 yeah,
non-conservative.
yeah, yeah. So,
Similarly, the last one, if you go to the last one, there are plenty of
yeah,
proline sitting and
proline and
one
tyrosine.
threonine or whatever it is, T.
Yeah, yeah,
So
yeah,
maybe that is also
yeah,
non-conservative. From that
yeah.
angle, maybe okay.
Yeah. So generally, when we talk about colons and semicolons, they basically denote that whether all of the bases are same or whether one is different or whether two are different. Okay, generally speaking, that is what happens. So maybe this is not a very good representation to have kept, but I will probably modify this before I send out the slides.
There is
Also,
one more, madam, in
yeah,
that one. Go back.
yeah, this
See,
one,
no, no. That one. Threonine,
yeah,
valine, isoleucine, threonine, they are
yeah,
conservative. That is not,
yeah.
that should be changed to conservative. Otherwise, everything looks okay to me.
Actually, this conservative and non-conservative is not really depend. Like as far as I know, when we talk about homology, we do not see whether these amino acids have similar functions or not. I don't think that is the basis for making a consensus or a non-consensus sequence. I think just looking at the sequence is basically used to say whether it is a consensus or a non-consensus sequence. So this is one thing that I will have to go back and check.
Sure, sure, no problem.
Okay, so I have picked another example of multiple sequence alignment also, wherein there are some accession IDs that are given. So I do not really know what these accession IDs stand for, but over here we have some scientific nomenclature that has been assigned to each accession ID. So if somebody is interested, they can come and look on this particular site. So why I have kept this slide is just to basically represent that multiple sequence alignment basically can align multiple sequences or for the same protein of same DNA segment between multiple different species, right? After it has basically done this alignment. For example, say for protein X, this alignment was performed and we found that this protein X was present in all these individuals, but some individuals had more similarity among the sequence maybe of this protein X than other individuals. For example, if we take these first two individuals, they are relatively closer to each other than this first individual and this last individual, right? So that basically defines the evolutionary distance between them. Also, once the nomenclature has been defined, we can see that the fungal species are close, have this protein X more similar to one another than the bacterial species, right? So bacterial species among themselves are more homologous in terms of this protein X, while fungal species among themselves are more homologous in respect or with respect to this particular protein X, right? So, in order to, okay, first let me just admit whoever is waiting. Yeah, Balaji, you can go ahead.
Yeah, the previous
Hmm.
slide, the right
Yeah,
hand
this one.
side, we have the phylogenic or whatever it comes, some kind of a tree is there. So
Yeah.
about
It
this
is a
tree,
phylogenetic.
tree.
Yeah.
So multiple sequence alignment will give this kind of thing. We call this as hypothesis. That may not
Yeah.
be fact in the sense,
Hmm.
if
Hmm.
two sequences are there, let us take an enzyme. All the things are perfectly aligned and beautifully there, except one amino acid residue in the active site. One may be functional, the other may be not functional. Multiple
Yes.
sequenced way, it may be totally beautifully aligned and then said, this is, close to each other. On the other
Yeah.
hand, there may be an enzyme where not from the active site, 2-3 mutations have occurred. So in the phylogenetic tree or this kind of
Hmm.
this tree, it may be far away,
Hmm.
but having the same function. So the statement, is this the statement? It proposes a hypothesis of similarity, not
Hmm.
definitive talk, definitive facts. That is the statement. Can that be made or not made? That is what I wanted to ask you. Unfortunately, I should not ask you this question because that is the question
completely all
number
right.
8 of
Yeah,
this week. Question
yeah.
number 8 of this week has that. So my
Okay.
understanding was that this multiple sequence alignment may throw a hypothesis that two proteins are very close to each other, but it may not have the same function
Yeah.
because only one residue in the active site may, topple its function. So just sorry to interrupt your throughput,
Yeah.
but just wanted to point out that. Thank you.
Okay. So there are actually two ways through which we can actually look at this. So I have not really gone through the current assignment, so I cannot be biased also to actually answer that question. Uh, but okay, we can maybe look at that also so that I can help you out specifically. But after we are done with this particular assignment, but two ways that we can look at this particular query is that what is the question that we are asking. For example, if we are talking about a protein X, it might be a protein that has already been identified in all these different species, fungal species as well as bacterial species, right? So it might be a already defined protein that functionally we know that it is present and performing this particular function in fungi also, and it is present and performing this particular function in bacteria also. We just want to understand that in like evolutionarily, how did it evolve? Was it first first did it come or appear in case of the bacteria, or first did it appear in case of fungi? So if that is the kind of problem, it can easily be solved using this particular tree. But if suppose we have discovered a new protein and we want to see, this is the second approach. We have discovered a new protein in say mice, and we want to see whether this particular protein is also present in humans. Like, why do we want to check that? Because we want to know that if we determine the function of this particular protein in case of mice, will it help? If we know that it is present in humans also, will it have the same function, right? So if a new protein was discovered, then it would be a valid hypothesis if we basically receive some kind of an alignment with the human genome also, we could say that, yeah, we think that this protein is also present in case of humans. So, but yeah, so in that case, it will be like a hypothesis, because we don't know that whether if even if this protein is present in humans, are we sure that it's just going to perform the same function that it is performing in case of the mice? So yeah, in that case, it could be a hypothesis. But like, what is your question is what is defining how you are interpreting this particular data, right? So, okay. Let me just finish with this phylogenetic discussion, and maybe we can go and check that question also.
Okay. There
Yeah.
are some numbers 6,500, 29, 4,700.
Yeah. So
What are
I
those
will
things?
explain what these
Okay,
are.
thank you.
Okay, yeah. On a very generic level, I will introduce this because this is one of the topics that is like it requires another whole session, right? So, but yeah, I will in a generic way, I will introduce everything that is present in this particular tree. That what does it mean, right? So first, before we actually talk about that tree, let us look at some MSA tools. For example, there were some examples that were already given to you during the tutorial session, one of which is Clustal Omega. It is one of the most widely used tools even right now. Other tools involve muscle. So there are some algorithmic differences between these. Okay, all of these I have taken from this EMBL website, and the website has a description about each of these. So if anybody is interested, you can go and basically scroll and through this website, and you will actually obtain a lot of information about these tools, right? So one just listing a few tools. They are Clustal Omega, Muscle, Tea Coffee, MAFFT, K-align, and other such alignments, right? So, okay. So now let us look at the phylogenetic tree and what it basically means. Okay. So let us first start a discussion with a simpler thing. So, for example, we are saying that let us start with species A. So, species A is one species and it is closely connected with species B, right? Then you see that there is a line that is connecting the two of them, and this line is going over here and joining with this other set of species, which is species C and species D, right? So, where the any two species are connected, any connecting point is basically known as a node. Okay, for example, if we take the example of a tree or a leaf itself, a plant has nodes and internodes, right? So nodes are places where the branch is connected to the main stem, right? And internodes is a different distance between different branches. So similarly, we have nodes over here, which are basically connecting any two branches. And the internodes is basically this distance between them, right? So this is one term that we wanted to introduce, that any two species will be connected by a node, and this node will be another maybe parent species from which these particular species have diverged. It could be possible, right? Other things that have been labeled over here are the branches. So you can see that species A, the branch looks bigger. In species B, it looks smaller. In species C, it is lengthy, right? In species D, again, it is a little smaller. So similarly, every species is having a different length of this branch, which is arising from the nodes, right? So these branches could have different length. What does this different length signify? It basically signifies the distance or the evolutionary distance, right? So for example, if we say that the distance scale is 0.01, right? So we say that if this much length is present in a branch, say suppose this much, so it is about 0.01 like 0.01 in terms of time scale, right? So it is basically an arbitrary unit. It is not a unit in years or kilometers or distance, any such thing. It is an arbitrary unit. It basically says that if we say from this particular individual, this particular A individual is 0.1 times distant, and this is 0.05 times distant. So we know that from this particular individual, species B is much closer than species A, right? Also, this particular distance can be used to calculate the distance between these two species also. For example, we want to know that how much evolutionarily diverged are species A and species B. So for this, we could basically calculate the branch length for species A, calculate the branch length for species B with whatever distance scaling has been given in a particular phylogenetic tree. And from that, we can say that the distance between these two is this distance plus this distance. Okay. So this is essentially, these were the things that were mentioned in this particular phylogenetic tree. Now, let us also just go through the definition of these things before we come into other types of phylogenetic trees and how they could be differently made, basically. Okay. So when I talk of a node represents a taxonomic unit. What are taxonomic units? They are units such as genus, species, family, phylum. Okay, such taxonomic units. This can be a taxon, which could be an existing species or an ancestor or an unknown species. For example, if it is not labeled, we might not know what it is, right? Oh, hello.
We can hear you. You can continue.
But can you also see the presentation?
We can see the Now the PPT has gone. Now we can see the fantastic. We can see phylogenetic tree.
Okay,
Slide
okay.
is
So
there.
you were not able to see it before this?
We can see topology, the branch pattern.
Okay, 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 okay.
We
Okay.
can
So
see this slide.
I will just resume my whatever I was explaining. So what I just talked about was node. So a node could represent one taxonomic group. This taxonomic group could be a species, a genus, or any such other taxon, right? And we might be aware of what this particular node is, or its identity might not be known to us. We might just know what species have diverged from here, but we might not have the knowledge about what they have diverged from. Okay, so it might be an extinct species, for example. Also, when we talk about branches, they basically define the relationship between the taxa in terms of descent and ancestry. For example, over here, we can say that species E has the same descent as species F, right? No, okay. This is actually incorrect because there is no node that has been connected over here. So we can say species E has same descent as species A, right? But species A, if we see the distance or the distance of the branches, we can maybe say that it is closer to its parent than species E, but that we will understand only when we know the branch length of all these branches and we can actually compare it to the branch length of species E. Okay, so it will basically defines the relationship between different taxa. Topology is basically the branching pattern. So over here we have this kind of a topology. Remember this term because we will be discussing about different kinds of topologies of different phylogenetic tree in the upcoming slide. When we talk about branch length, right over here, for example, this branch length, it often represents the number of changes that have occurred in that particular branch. But okay. When we talk about the root, it is the common ancestor of all the taxa. For example, species F is the common ancestor, like common relative of all these species, right? Species A and species E might not directly be related, but species F is where every other species is coming out from. So it is a common relative of all the other taxons. If we talk about the distance scale, it represents the number of differences between sequences. For example, 0.1 over here represents 10% differences between two sequences, right? So for example, this roughly is equal to this. Okay, so we will say that species A is 10% plus this looks like half of this, right? So plus 5%. So 15% difference is there between species A and species B. When we look at the topology or the different ways that a phylogenetic tree could actually be drawn, we can say that basically when we talked about a root over here, so a root means we know one parent. Okay, for example, if I was to talk about myself, I would consider in my family the root to be my grandparents, right? From my grandparents, my parents have my father. For example, if I talk about my paternal grandparents, they give birth to my father and my father's brother, right? Then my father has his own family and my father's brother has his own family. Now I am a part of my father's family and my cousin is a part of my father's brother's family. But we are basically connected to each other and we know the point of connection or the root of connection, and that root is our paternal grandparents, right? So in some cases, we are aware of what the root is. But sometimes what happens is that we know that two people or two species, for example, are connected. But we don't know how they are connected. Or we don't know that if, for example, we were talking about a particular kind of algae and a particular kind of fungi. We say that these are evolutionarily connected. But we don't know whether this particular kind of algae came first, or this particular kind of fungi came into existence first, right? So we don't know which one is the one that is preceding the other one, right? In that case, we basically make an unrooted tree. In this particular unrooted tree, for example, we can see that uh, if you generally just zoom in and look at this particular. Okay, let me just get a zoomed in view. Yeah. If you just zoom in into this particular space, you will not be able to basically tell which one is the root, right? For example, did species A come first, after that species B is coming, or species E came first, after which all of these species are coming. Like, you are unable to explain that in such a phylogenetic tree, and such trees also exist, right? For example, in this other example, in this other one also, it is you will be unable to tell whether species B has come first, after that species A has diverged from it, or has were species E coming first, from which this D and C have basically diverged. So you know that they are related, but you don't know how they are related or what precedes the other one, right? Then again, you can appreciate the difference between these two trees themselves, right? So in the first case, you see that the branch lengths are very, very different, right? Some where the branch is very, very long, or somewhere the branch is very short, while in the second ones, the branch lengths are same across all the species, right? So the second case is a case in which the branches have not been scaled, right? This means that they have just shown that these species are connected, but they have not shown that how much they are connected with each other. For example, in the first one, wherein the branches have been scaled, you can see that species A and B are closer to one another than species A and E, right? Or species C and D are closer to one another in, say, species C and A, right? Because you can see that there is different distance that is there between these species. But that is not always the case in case of these unscaled branches. This does not basically give us the distance or the accurate distance between two species, right? After that, there is another kind of topology in phylogenetic trees, which is basically, now I'll describe what these scaled branches are and what unscaled branches are, right? So first, we describe that it is not always necessary that we have a root, right? But in cases that we do have a root, right? For example, this other example that I am showing over here, in this case, we do have a root in both these branches, in both these trees, right? But in the first one, the representation is a little different than the second one, but both of them are scaled, right? So the unit is written over here and both of them are scaled. So they can still tell that how much exactly species B is diverging from the root, how much exactly distance is present between species C and D, and same is the case for this particular tree on the right hand side, right? So over here, they are basically giving the same outcome, just the representation is different. And you might have seen when you look at phylogenetic trees, a lot of time you see such arrangement wherein these boxes are drawn, or sometimes you see these diagonal lines only that are drawn, right? Then there are also unscaled branches, like we talked about in the first case. So these unscaled branches will basically represent the divergence of species, and it might even represent the which one is preceding, right? Which one is preceding which. So over here, we know that the root species is one that is mentioned as root. After that, which is coming first? Maybe. Okay, so yeah. So over here, actually, in unscaled branches, after the root, you will not be able to tell which species is coming first. But over here, you might be able to tell that after the root, species D has lesser distance from the root than species E. So maybe species D is coming first and then species E is arising. Okay, so there are different kinds of phylogenetic trees that are actually present. It is a very, very diverse topics, and there are multiple tools that are used for creation of these phylogenetic trees also. Clustal is a basic multiple sequence alignment tool that gives the data for such an alignment, right? So that is why we had discussed this briefly. If at all there is any query, particularly in phylogenetic trees, or how exactly it is built that you want to know or understand, so maybe I can include that in the upcoming sessions. Okay. So moving on to our fourth question, which basically says, array-based comparative genomic hybridization, or array CGH, is a popular method for analyzing whole genome content. Which one of the following cannot be detected using array CGH? Is it balanced chromosomal translocations, or copy number variations, or copy number amplification, or all of these? So in order to understand the answer of this particular question, we basically need a basic understanding of the principle of array-based comparative genomic hybridization. So it is somewhat like microarray only. So since you have briefly read about microarrays, I urge you to go through the question once and we'll come to the answer. Okay, so in this particular question, the answer is balanced chromosomal translocations. So array CGH can identify the quantitative differences in the genome. Okay, so quantitative differences such as how much a particular gene is amplified, how much copy number variations are present, or if there is an insertion or deletion, but it cannot assess qualitative changes such as balanced translocations or single nucleotide polymorphisms, and it cannot validate these things. So how does this array CGH actually works? For that, again, I have basically pasted a very simple workflow that I have sourced from Agilent, which is one of the companies that basically provides the services for this particular technique. So over here, the first step involves extraction and labeling of DNA. So since it is comparative genomic hybridization, means we are comparing two kinds of DNA, right? So therefore, there will be a control or reference sample and there will be a normal sample, whatever we want to test, right? So first we will extract these two kinds of samples and we will label the DNA. So extract genomic DNA from a test and a reference sample and label one with a red fluorescent dye and the other with a green fluorescent dye. Okay. After they have been labeled, what we will do is that we will leave them to hybridize and wash. Right. So first we will basically take this array plate or array chip, wherein some kinds of oligos or material will already be present on that chip on which these particular samples will go and hybridize, right? So once they hybridize, we will give some kind of washing to this particular chip or slide. And why will we wash it? So that whatever is unbound will be washed away. So we will mix and hybridized to a microarray printed with a thousands of oligonucleotide probes, and then we will wash it. So over here you can see that if there could be equal hybridization, that the control sample goes and hybridizes, one of the control sample hybridizes and one of the DNA sample hybridizes, or there could be DNA dosage gain where more of sample DNA is hybridizing and less of control DNA is hybridizing, or there could be a loss wherein more of control is hybridizing and less of sample is hybridizing. And then we will basically detect these changes via scanning this. Okay, so we will detect the red and the green signals using a fluorescence scanner, and then we will analyze this data using different tools. such that we compute and report the gains or losses in the test DNA using a software. So if we pictorially had to understand how it would look like, it will basically, when we mix the two DNAs and we put it on the DNA array plate or microchip, what will happen? It will hybridize at different locations. Since the patient sample is labeled green and the reference is labeled red, wherever only green is coming, that means that only this patient sample is showing expression. Wherever only red is coming, it means only the reference sample is showing expression. Wherein we have yellow color, it means that there is equal expression of both the patient and the reference. So just a moment, let me admit whoever is waiting. Yeah. Did you also want to ask a question? Okay, it was a previous one. I think I will just share my screen again. Yeah. Okay. So, wherever the expression is same between the patient and the reference, we will basically obtain a yellow color. Right, so a yellow spot basically means a 1:1 ratio. A green spot means that there is more number of green hybridization or patient DNA hybridization than the sample DNA hybridization, and the
We
vice
cannot
versa
see
for
your
the red
slide.
spot.
We cannot
Oh,
see
you
your
can't?
slide.
Did it happen just now or has it been so for a while?
Now again, let's say, now we can see your face. Now we can see.
Okay, were you able to follow this particular slide? Did you know I was on the slide?
Yes, you were talking about this slide only. Yeah.
Okay, so okay, I had talked about the brief workflow of the array CGH, and I was talking about how it would look like if we had to pictorially understand it. Okay, so for example, whenever the computer will be analyzing this particular slide or this particular chip, what the computer will do is that it will basically look at the fluorescence that it is emitting, right? For example, if there is any gain or if there is higher expression of this green or the patient sample, then there will be an excitation over here that will show green emission, and the computer will read it as a more number of copies may be present in case of the green or the patient sample. If there are losses or deletions, then the red will show diverge, and since there are not equal copies that are present, so instead of yellow signal, it will give red signal, and that will mean that there is some kind of a deletion that is present in the particular patient sample, right? So this is roughly how we assess the patient sample using a reference sample via array comparative genomic hybridization.
Excuse me, madam.
Yeah.
In the previous slide, patient sample means, let us say, disease patient sample. Control means normal person, is it?
Yeah.
Okay.
So there could also be a library of these normal samples that these labs might have. So maybe they work on that, or maybe whenever a control sample is taken, for example, if I went for a screen of my genes, my DNA, a quick screen of my DNA, right? I don't want to wait for the sequencing, or the sequencing costs too much. I don't want to go into sequencing until and unless I know that, yes, there could be some problem with my DNA itself. So I give my sample, and either the company might already have a library of control samples, or they might just ask that, okay, I am taking her DNA. Can I also take one of your family members' DNA who is not facing that same problem, right? So that could also be a control sample. Okay.
How to decide on what should be there on the chip? What kind of complementary oligos should be there?
Yeah. So what happens over here is that there are chips that are there are different kinds of microarray chips that are available now. So there will be some, for example, if there is a cancer patient, right? So you will basically just have to assess the major cancer genes that might go haywire, right? So you will basically create a microarray chip which will basically encompass whatever cancer genes you can actually you generally see mutations in, and you can actually basically fit them in that particular chip. For example, if there is an individual who is having maybe some kind of a dementiated condition, right? So that individual will basically be assessed using a chip that has the mutations that are basically mutations that people assess when there is a neurodegenerative condition. For example, there will be genes relating to Alzheimer's, Parkinson's, and multiple epilepsies that will be put on that particular chip, right? So it will depend on which kind of patient you are basically treating. So if you are an oncologist, for example, oncology also involves multiple different disciplines, right? So somebody might be a breast cancer specialist, some other person might be a prostate cancer or colon cancer specialist. So they will probably assess like if you suspect a particular kind of disease in an individual, this is one of the prognostic tests that you can do to understand whether there is some genetic level defect also that is present. Although in clinics now, arrays are not that commonly used. Sequencing is the way to go. But yeah, on some superficial level, like they cannot give you deterministic results. Or, for example, a lot of cancers involve translocations, and we understood that if the translocation is like, if there is what is that kind of translocation? Reciprocal translocation. If that reciprocal translocation is present, you cannot detect it via these techniques, right? So sometimes it is useful, but on a very superficial level, it is actually used in terms of clinical relevance. In labs, it is still a very good technique to screen out a lot of things.
Sorry to disturb.
Yeah.
Stupid question. When you
No,
say
no, no.
patient and reference samples, is it DNA or RNA?
So there are different kinds of
mRNA.
arrays. So there are
Which one
different
we
kinds
take?
of samples actually. Usually it is cDNA only. Like mRNA is extracted, made into cDNA, and that is checked in a DNA chip. Now we also have protein arrays actually
Very good.
to directly check for proteins.
So protein array means how it is going to hybridize with the spots on the microarray. Is it going to have an antibody or what is that?
Yeah. So proteins will have different kinds of oligos that it will hybridize to. Like these DNAs are not the oligo, the basis for them to hybridize are not going to be the same. They will probably contain other proteins with which to go and attach.
Thank you, madam. Thank you, madam.
Yeah. So coming on to question number 5, we have shown here is a schematic of an assay that is used for genomic approaches. Which one of the following, which one of the option correctly identifies the method that the scheme represents and its objective? So the first one is fluorescent in situ hybridization, which is utilized for location of genes on chromosomes, or Western blotting, which is used for protein estimation, or chromatin immunoprecipitation, which is used for chromatin dynamics, or in vitro DNA synthesis, which is used for DNA amplification. So this is a very similar schematic to what we have already discussed. So not wasting much time, I'm going to the answer, which is chromatin immunoprecipitation, which is basically used to study the dynamics of chromatins, right? Okay, first I will introduce the next question, which is which one of the following options is not an essential prerequisite for shotgun sequencing approach? Is it the presence of DNA polymerase or a host vector or information on the position of DNA fragment in the genome itself or the formation of a DNA library? So which one of this is not really essential when we are doing shotgun sequencing? Also, please excuse me for a moment. I'll just shut this moment. Yeah. Okay. So shotgun sequencing approach is one of the approaches that was used for DNA sequencing, right? When we look at this, the prior information on the position of the DNA fragment in the genome is not really important, right? Even if we know it, we can fragment the DNA accordingly. But even if we don't know it, we can still basically sequence the DNA using this particular method. So what this method involves is basically the presence or extraction of a genomic DNA from an individual or a model or any cell, right? After that, we create a library, and this library is usually created inside the or the uh, bacterial artificial chromosome library is created. So this is basically where the host vector comes in, right? After this library is basically created. Why is it first of all created? Because we might not have the knowledge of the individual segments of genes. So we want every segment to be incorporated in one of the back libraries. And yeah, just all right. After which these basically will be cloned and they will be like this contiguous explained, right? Continuous sequences will basically be created. So these sequences will basically be screened out via different automated tests only. And then the overlapping sequences will be looked for and contexts will be created so that we have a basic outline of what kind of a sequence it will form, right? After that, we will basically transform them into shotgun clones, and sequence will be done one by one, right? And such a result will actually be obtained. So for this, we will actually need a DNA polymerase because we need to synthesize these clones, right? Then we will need the host vector in order to create a library when we don't know what these DNA fragment, where this DNA fragment will be lying, or even if we don't know about the sequence of this particular fragment, right? And then we will also need a DNA library. Why will we need a DNA library? Because even when we are mapping these sequences through this overlaps of in these repeats, there might be some gaps that we will obtain in between, right? So for that, we will need a DNA library to fill in those gaps. But prior information on position of the DNA in the DNA fragment in the genome is not really a prerequisite for shotgun sequencing. Thereafter, we have another kind of a chip that is shown here. And the question is, shown here is a schematic of an assay platform used for genomics approaches. Which one of the following option correctly identifies the assay depicted and the property of the probe indicated? So this is an Affymetrix chip. Affymetrix is actually one of the companies, and there are certain probes that have been defined over here. So they are asking that what kind of a chip is this, and if you have identified this chip, then what kind of a probes will these be? So coming to the answer, this particular chip is a gene chip. Okay, so it will basically identify multiple genes. And the probes that are used over here are oligonucleotides that are complementary to the mRNA sequences or the target DNA or to the target DNA, right? And they are not fluorescently tagged. So they will not be fluorescently labeled DNA, they will be oligonucleotides. What happens in a gene chip actually is that we again have a microarray like chip, right? Wherein we will basically give some sample, which we will extract sample first of all, then we will extract maybe, okay, somebody's trying to join. Yeah. Yeah, so in this case, we will basically extract sample, then we will extract the mRNA out of that sample and we will fluorescently label it, just like we used to do in case of a microarray. Then we will leave that sample in this gene chip and we will let it hybridize. And after it hybridizes, we will let a system that is attached to this particular machine to actually detect the expression level of various genes, right? Uh, the expression level of various genes can be identified. Uh, okay, there is somebody trying to join. I'm so sorry for the other learners. Yes. Okay. This is one of the examples of a gene chip by Affymetrix itself, which is a human transcriptome array. Okay, so in this, this is one of the chips that holds complementary oligos to human DNA, right? And it can basically be used to check for some alterations in sample DNA. And this is what the data actually looks like when we look at a gene chip. If we zoom into one of this inset, we see that this is how it actually looks like, which is actually read by one of the analyzer or analyzing machines. So it will either give this yellow colored result for it will be bright green if there is expression only in the this patient sample, or it will be red if there is some kind of a down regulation of a particular gene in the patient sample. So this is how it looks like. Moving on to question number 8. Based on your understanding of gene sequencing technologies, identify the type of gene sequencing given here in the schematic. So this is one of the schematics that have been given, and they are asking whether it represents pyrosequencing or Sanger sequencing or Illumina sequencing or none of these. So which kind of a sequencing do you think this is? Since it has this PPI, that pyrophosphate that is mentioned over here, you can easily recognize this is a pyrosequencing. It is basically a method of DNA sequencing based on sequencing by synthesis principle, in which the sequencing is performed by detecting the oligonucleotide incorporated by the DNA polymerase. The pyrosequencing relies on light detection based on chain reaction when pyrophosphate is basically released. Okay. So moving on to our penultimate question for the day, which is which one of the following options is incorrect with regard to difference between Sanger sequencing method from the next gen sequencing method? So is it that Sanger sequencing is targeted to a segment of DNA template while NGS could sequence the whole DNA template, or NGS method can sequence hundreds to thousands of genes or regions simultaneously, while Sanger method could sequence only a part of the gene per run, or Sanger method is not scalable while NGS method is scalable, or Sanger method is cost effective while NGS is expensive. So which one of these do you think is the incorrect statement with regard to both Sanger sequencing and NGS sequencing method? So it was a fairly apparent one, which is why we are not pondering much upon this question. And the answer actually is that Sanger method is cost effective while NGS is expensive, because this is vice versa in the actual case, right? The others are true because the first one that says that Sanger is targeted to a segment of DNA template while NGS could sequence whole DNA template is absolutely true. The second one is an extension of the first statement, which says that simultaneously NGS is able to run thousands of sequencing of thousands of genes, but in capillary we are running it through a gel or a capillary method. Only one sequence at a time we can actually obtain. And because of these two things only, Sanger method is not really scalable. For scalable in the sense, if we had to perform it on a large scale, will we be able to do it like as efficiently as NGS? No. NGS is much more scalable. For example, even if we have a large number of patient samples, it is easier to perform NGS sequencing than Sanger sequencing in that patient data. Okay. So, question number 10 says, a BLAST is commonly used for which one of these? Constructing phylogenetic trees for organisms based on their sequence diversity, or organizing biological information around the sequences of large genomes for different species, or comparing primary biological sequence information such as amino acid sequences of proteins or nucleotides of DNA and or RNA sequences, or all of these. So which one do you think BLAST is actually used for? The answer being the third one, which is comparing the primary biological sequence information such as amino acid sequences of proteins, nucleotides of DNA or RNA sequences. So BLAST basically is like if we expand it, we can say BLAST is basic local alignment search tool. So its primary function is alignment, right? So it finds regions of similarity between biological sequences. The program basically compares nucleotides or protein sequences to sequence databases and calculates the statistical significance of this data, right? Okay. I have again mentioned this particular screenshot of from the National Library of Medicine, which basically contains the BLAST local alignment search tool also, basic local alignment search tool or BLAST. And that is it for this particular presentation. And we will jump on to the next one because we have actually spent a lot of time on the Q&A this time. Okay. So this is presentation number 2 for today, wherein we discuss about some of the other questions and concepts that we might have had pertaining to this week's lectures. Also, it does contain some part of what were remaining questions from last week's discussion, right? So that is also what I have explained. I think, yeah, I have actually begun with that. So last week, what happened was that we had a fairly long discussion about what exons are, right? Because why was this discussion very long was because I don't know if the particular, I think Ajay joined, but he's not there in this meeting. So I will explain it to the other people what this discussion was. So this discussion basically was that we talk about alternative splicing when it comes to exons and introns, right? Forming mRNA. We talk about alternative splicing. During splicing, introns are the sections that are spliced out, and the exons are basically joined together. But during alternative splicing, what happens is that sometimes even the exon skipping occurs, right? Or there might be exons that are exclusive of exclusive of one another that might be present. For example, if we say, if we look at the exons, what happens is that what are exons? They are coding sections of an RNA transcript or the DNA encoding it that are translated into proteins, right? So the definition of exon is that it should be coding a protein, right? The human genome contains about 180,000 exons, which are collectively called as an exome. Okay, so for example, the collective mRNA of the entire organism is called a transcriptome. The collective genes are called a genome, right? Similarly, the collective exons of an organism are known as exome. An exome comprises only about 1% of the human genome. So it only comprises 1% of the human genome. So only 1% of the human genome is actually coding for proteins. So what happens during transcription and during transcription is basically that there are DNAs that are present. These DNAs have regions of these protein coding exons, and between them they have regions that are not coding for proteins, right? These are known as introns. The ones that are not colored over here are introns. So when transcription actually happens, during the pre-mRNA stage, wherein the mRNA has not actually matured, this particular stage will contain both these exons as well as these sequences that are not coding for the proteins, which are known as introns, right? What will happen after this is splicing will take place. During splicing, the introns or the non-coding regions are actually removed out, such that a mature RNA is formed, which is known as an mRNA, which can basically go on and it can translate into proteins, or it can instruct the formation of proteins, right? But what also happens is that this is not the only case. Sometimes there might be shuffling between these exons that might be occurring, right? Okay. Yeah. So let me just start my screen. Yes. Sometimes shuffling might be occurring during between these exons, right? So what would happen is, say, suppose exon 1 is present in the mRNA, along with this, exon 3, 4, and 5 are present. Exon 2 is not present. Or in this case, for example, exon 2, 3, and 4 are present. Exon 1 and 5 are not present. Sometimes these cases might arise. Okay, so these are outcomes of alternative splicing. So last time, one of the discussions that we had was that if sometimes the exon is also not coming and forming a mature RNA, how can we say that it is an exon? If it is also being spliced out like the introns, why do we say that it is a exon? Right. So I tried explaining, and I am going to try explaining in almost the same along almost the same lines today as well, with another example. Okay. So in case of exons, what I answered last time was that exons are coding for proteins. It might be possible that a particular mRNA in which they are not included is forming some other kind of protein, but also this particular DNA is forming multiple proteins, right? For example, protein A, B, C, and D. So maybe a particular exon 5 is not involved in coding of protein A, B, and C, but it is performing a function in encoding protein D, right? So since it has some role in protein coding still, maybe not in protein A itself, but in another protein, it is still known as an exon, even though it is not coming into that particular mature RNA. Right. So, just to explain that, I kept an example of a similar concept, which is multiple allelism. Now, this is a concept that I am bringing from a very different topic, which is basically genetics. Right. So, in genetics, let me briefly introduce this so that you can follow this discussion. What happens is, if anybody recalls what multiple allelism is, multiple allelism basically means that there could be one or more than one alleles of a gene that could be present. Now, I hope you guys remember what alleles are. For example, if we say, okay, I need to annotate. Yeah. So, for example, if we say that, okay. Right. Yeah. If this is a particular gene, if this is a particular chromosome, right? A chromosome has two chromatids, right? If we actually spread out these two chromatids like this, right? Consider them to be similar in size, shape, and all these things, right? These two chromatids are spread out. Now, they will basically contain two forms of a gene which are known as alleles. So this is one locus and this is the same locus on the other chromatid, right? So they will contain two genes. Now, they will contain the same genes, but one copy will have come from the father and the other will have come from the mother. Okay, for example, this is one chromosome. This is one chromosome that we are talking about, and we have taken one chromatid from this chromosome, and there is another chromosome we are talking about, and which has come from the mother, and the other chromatid we have picked from the mother chromosome, right? So one is from the father chromosome and the other is from the mother chromosome. Now they both have the same gene. This gene is responsible for our blood group. Okay, but one could either both the genes could have the same alleles. Say if both the genes have the same allele, which is A, right? Or same form of that gene, then it would give blood group A. If they had the same allele, that is B, they would give blood group B. Or if they had different alleles, that is A and one had A, the maternal had A and the paternal had B, it will give blood group AB. Or there could be other forms of different difference that is present. Wait, let me add. Okay. There was somebody trying to come into the meeting. Anyway. Or say if one strand has allele A and the other has allele O, right? In this, since the A1 is dominant, the blood group of that individual will still be A. But I hope if somebody wants me to teach this concept, I can teach it in a more simpler way. But I am assuming, so please stop me from my assumption if you need to actually know what this is. So I am assuming that you guys might be familiar about these this concept of alleles, right? So alleles is basically different forms of a same gene, right? And different alleles, if they are present, they could give different outcomes in an individual, right? Also, one particular individual cannot have more than two alleles because they have just two homologous chromosomes, right? So they have just two copies of that gene. One is coming from the mother, one is coming from the father. So that particular individual can have just two copies of a particular gene. But if we look at the population, it might be possible that there are more than two copies of that particular gene that are present, right? How is that possible? For example, if we talk about this blood group again, we say that one individual has blood group A, the other one has blood group B, and the third one has blood group O, right? It is possible that one individual has two of these three alleles, right? Somebody could have A and O, that person will have blood group A. Somebody could have B and O, that person would have blood group B. Somebody could have A and B, suppose. That person could have blood group AB. Somebody could just have O in both the genes, and they will have blood group O, right? But is there any individual which will have all three kinds of alleles, blood A, B, and O? Can any individual have three copies of an allele, a normal healthy individual? So the answer is actually no. So like we talk about such conditions about multiple alleles or polymorphic genes or polymorphism as a concept in a population rather than in a particular individual. Similarly, when we talk about exons, we cannot talk about one particular mRNA. Okay, we have to consider it as a particular genome or multiple proteins. Okay, so in multiple proteins, that a particular exon might be encoding for some region of some protein, right? But it is not important that every exon is coding for every protein that that particular gene is responsible for making. So since the person who actually asked for the query is not here, I will not know whether or not I have resolved it. But I have basically written this down also in my own words. So for when you actually receive this presentation, so that you can understand what I was trying to explain earlier. Okay, so that brings us to an end of this conflict of what exons were and why they, even though they were not present in case of some mRNAs, they were still known as exons, right? So, okay, after this, I come into another concept that I had prepared for last week, but I was unable to explain it due to time constraint, which is basically repair mechanisms. So last time we talked about some repair mechanisms. We did not really talk about repair mechanisms, and the course actually touched upon repair mechanisms when they were talking about homologous recombination, or how sometimes homologous recombinations are basically using the host's own native repair mechanisms to actually recombine some new genes, right? So for that, I wanted to lay stress upon superficially what some of the DNA repair mechanisms are that exist in our body. So in our body, we largely have 5 kinds of DNA repair mechanisms. One is the base excision repair, which is usually when there is a single-stranded base, and usually a uracil is involved over here. Okay, and it usually occurs because of oxidation of the DNA. So when reactive oxygen species are present, it might lead to a single-stranded break due to some kind of a stress in the DNA, and it will basically call for a kind of excision repair, which is the base excision repair. Then there is another kind of a repair, which is mismatch repair or MMR. Now, this basically corrects AG mismatch or TC mismatch, or it could even correct for insertions or deletions. Then again, there is a third kind of repair mechanism, which basically checks for bulky adducts or intra-strand crosslinks. So, if such kind of crosslinks are ever present in between strands, or if there are heavy additives that have been basically present are joining to the DNA, then nucleotide excision repair basically heals these conditions in case of a DNA. The final two are the ones that we are actually going to discuss in detail, which are double strand break, interstrand crosslink, wherein there is a double-stranded break. And for this, 2 kinds of a repair are used. Both of them are under this category, that is double-strand break repair. One is the non-homologous end joining, and the other is the homology-dependent repair, right? So over here, we are going to focus on these two. Since functional genomics, a lot of the techniques that are involving recombinant DNA technologies, they are actually using the host native non-homologous end joining and homology dependent repair. So what are these two? So as we discussed, they will basically be repairing double-stranded breaks, which is labeled as DSB over here. What happens is that whenever there is a double-stranded break, there are 2 kinds of pathways that the DNA could take, right, for its own repair. In the first case, what happens is in non-homologous end joining, there is a protein that is crupen, which comes and identifies that there is a double-stranded break, right? Once there is this identification, it will basically recruit these PKCs or protein kinases on this particular segment, wherein on the side, both sides of the segment, wherein this particular break is occurring, right? After that, there will be a nuclease that will come into picture, which is Artemis. Now, what this nuclease will do, it will further trim this particular break, right? So it will trim the break such that there are some kind of complementary segments that are basically now visible between these two different broken segments, right? After this nuclease has done its activity, this complex that involves LIG4, XLF, XRCC4. This particular complex will come and bind to these two strands, and it will basically cause these strands to stick together, or it will cause a ligation activity. So, thus NHEJ will basically result in some kind of insertion or deletion, usually deletion. So, while it is repairing such double-stranded breaks, right? The other kind of DNA repair mechanism is the homologous recombination or homology-dependent recombination, HR or HDR. Now, what happens in HR is that whenever there is a double-stranded break, if this particular mechanism has to be healed via a homology dependent recombination, what will happen is that the proteins that will come and bind over here are different, mRNA and CTIP. Now, when they come and bind, what they will do is that they will further basically block the binding of Crewell, for example, that will basically recruit for NHEJ like repair, and they will also open the strand up a bit. Now, once this opening has occurred, it will no longer be able to be basically repaired using non-homologous end joining after the gap has spread after a while, right? So there will be other proteins that will come and basically lie on these naked strands, which are RPA proteins. Now these RPA proteins will eventually be replaced by RAD51 protein by the function of this BRCA2, right? After this RAD51 take place of this RPA protein on this naked DNA strand, what will happen is that they will aid its invasion into a sister chromatid, right? So there will be a sister chromatid that will be lying close to this. So because we are talking about homologous chromosomes, right? So there will be another homologous chromosome, and it will basically take this particular broken DNA to that particular homologous chromosome, right? After it has taken it there, it will use the sequence of this particular chromatid to basically rebuild or synthesize the leftover remaining or missing part of the strand. After that, what we will receive after this process is two complete DNAs, wherein such a deletion has not occurred because it has basically used homology from its adjoining sister chromatid to basically rebuild what it already had and it had lost due to a double-stranded break. Okay, so based on this, NHEJ is active throughout the cell cycle and it does not rely on a template. But since homology dependent repair, it relies on a template strand to actually rebuild the DNA that has been lost. It is restricted to the synthesis and the G2 phases of the cell cycle due to the requirement for the sister chromatid as a template. Okay. All right. So that was it for DNA repair mechanism, specifically NHEJ and homology dependent recombination. As this was more extensively talked about in our functional genomics course. After this, another thing that we talked about extensively was the Cree-lock system. Now, what was Cree-lock system used for? It was basically used to, again, make recombinants, okay, or make inducible systems. So, Cree is basically short for Cree recombinase, which is one of the tyrosine site-specific recombinases or TSSRs, which was discovered first from a bacteriophage P1, right? It recognizes the specific DNA fragment sequences called LOXP. So these are particular sites that are recognized by this recombinase enzyme that is known as Cree. And when this Cree recognizes these sites called LOXP, which is basically short for locus of X over P1 site. It mediates site-specific deletion of DNA sequences between two log-P sites. Okay. So what happens over here is, in a 3-log system, when a Cree recombinase, which is a 38 kilodalton protein, when it is introduced, when the region that is present between two LOXP sites is basically deleted, right? So it will be deleted, and this particular Y region will be lost in our resultant gene. Also, what happens sometimes when we basically further correct or further improve this technology, which is cross log 3-log space system, what happens is that we can
Blackout. Hello.
yeah.
Your screen is blackout for I think 20-25 minutes. I don't know about others, but I cannot see your screen. It's totally blackout.
Actually, I can see it.
Is that the case for everybody?
No, no,
Oh,
we can. I can see,
okay.
madam.
Okay. Yeah, I don't know who raised the query. Can you please try to rejoin?
I can see, madam. I can see, madam.
Okay. All right. Okay. So there is another kind of utility of this pre-log system, which was also described in one of the sessions, which is conditional mutation or general breeding. So in this, what happens is that, again, the gene of interest is basically flogged by these LOXP sites. So once these LOXP sites are attached around the gene of interest, what happens is that we can basically let this particular individual. Okay. Yeah. So we can let this particular individual normally grow. And once this individual will grow, we will basically induce the activity of Cree. So this Cree is not normally given to just active in case of these individuals in which this kind of breeding is done. What happens is that the Cree is basically expressed with a tissue specific promoter. For example, if we wanted to target the thyroid gland, and we cannot really target the thyroid gland in a embryo, because if we had this particular Y gene deleted in an embryo in the thyroid gland, then what would happen is that the individual would die. But what we want to do is that once the individual grows, after the developmental stages have been completed, and now the individual is has been born and it can grow, after that what will happen is what will happen is that we want to, after the individual has attained a certain age, we want to take out this gene and see then what happens in that individual, right? So what we will do is that we will basically attach a promoter in front of this Cree recombinase, which will basically be active only when the individual will be like, it might be a promoter for a specific protein that is expressed only after a certain age in an individual, right? So we can basically conditionally cause mutation in that particular individual. So only after this particular protein is expressed in the thyroid of an individual will this particular Y gene be deleted in that individual. So this will help us attain our desired mutation, right? Also, there were other inducible Cree-lock systems that are available. For example, the tamoxifen inducible Cree system, which is basically achieved by modified Cree protein, which is fused with the estrogen receptor containing a mutated ligand binding domain. So thus, it is also known as a ER-LBD, okay, or Cree-ER, because it is fused with a Cree protein. And this is basically induced by one of the chemicals, which is tamoxifen. So what happens in the system is that now we have basically fused this Cree recombinase with this ER or estrogen receptor. And whenever we will actually give tamoxifen, what will happen is that this ER receptor, which can actually recognize tamoxifen, will get activated, and it will be able to come inside the nucleus. Along with it, it will also bring this Cre recombinase inside the nucleus, and now it can act on the LOXP sites that are already present inside the nucleus, right? So in the normal condition, the ER, since it is it has been fused with Cre, it keeps the Cre out in the cytosol. In the cytosol, the LOXP flogged DNA is not present, so the Cre cannot act. But once the tamoxifen is given, the ER is activated. This Cre can actually go inside the nucleus, and now it can cause the inactivation of whatever, or the deletion of this Y gene that was already present inside the nucleus, right? So that is why it is inducible. Only when we are giving this particular chemical will the system work. Okay. Also, there are other examples of inducible Cre-Lox systems. Other examples contain temporal and cell-specific inducible systems, such as tetracycline systems, which also sometimes instead of tetracycline use doxycycline. Thus, they are either called TET systems or DOX systems, right? These could be TET on systems or TET off systems. So what happens in these systems is that they consist of three elements. One is the reverse tetracycline control transactivator, which is present in the case of TET on systems. Tetracycline control transactivator that is present in the case of TET off systems, and tetracycline response element, which is TRE, or tetracycline operon, which is TETO, which is basically what is regulating the activity of the genes below, which is coming downstream of this particular operon, right? So what will happen in these TET on and TET off systems is that in case of the TET on systems, usually what happens is that this RTTA, that is reverse tetracycline control transactivator, is present in the cell, right? And when it is present in the cell, it cannot really go and bind to the tetracycline response element. And since it is not binding to this element, it is not starting the Cre recombinase. It is not activating the Cre recombinase. Since there is no activation of Cre recombinase, the expression of this particular floxed gene is still continuing. Now what happens is when tetracycline or doxycycline is given to introduced in the system, what happens is that it binds to this RTTA element. Now this RTTA can go and sit on the TET operon or tetracycline response element, and it activates the Cre and the Cre expression. The Cre recombinase expresses, due to which this particular gene expression is lost. Okay. So this is how the TET on system works. Now, just contrary to this is what happens in TET off system. In TET off system, instead of reverse tetracycline control transactivator, we just use tetracycline control transactivator. Now, what it does is that when it is present, it will basically turn on the activity of Cre recombinase, and Cre will basically lead to expression. But if we introduce tetracycline or doxycycline into this system, what happens is it inactivates this TTA, and it cannot bind or go and activate the tree, which will eventually not let the Cre recombinase express, and the further copies of this particular gene will not be hampered. Right. So this is another kind of inducible Cre-Lox-P system. So these are the three Cre-Lox-P systems that we discussed. Okay. So, okay, if we have time, we can just go through this also. I think this concept I can discuss. It is one slide. After that, it is not that important also. So this was another thing that I wanted to discuss in the previous class itself, which is gene knockout versus gene silencing. So what is the difference? So somebody who has attended, all of you have attended the course sessions, and some of you have also attended previous sessions with me. So would somebody like to speak up? What is the basic difference between gene knockout versus gene silencing? I will obviously discuss it. But if any of you would like to pitch it.
Gene silencing is something temporary and knockout is permanent.
Okay. Okay. All right. So the difference that we would like to start with is that gene knockout is actually interfering with the gene. But gene silencing is just interfering with the expression of that gene. Okay. So how it works is that in case of knockout, what happens is that a particular gene is present in a particular region on the chromosome and or a particular region on the DNA, and that particular gene itself has been targeted such that we are either removing that gene or we are introducing something else in place of that, such that somehow that gene is lost. Okay, so this is basically interfering with the DNA itself. Okay, and it will cause a knockout of the gene because we are removing a part of the DNA that was basically doing a particular function. But what will happen in case of a gene silencing? Over here, we will not touch the genomic DNA. Okay. Instead, what will happen is that the expression of this particular DNA will be hampered. Now, how does it express? First, through transcription, it will express at the level of mRNAs, right? Then through translation, it will express at the level of proteins, right? So either this transcription is somehow blocked or mRNA is somehow blocked to basically form a protein or form a functional protein. That is basically how gene silencing is done, right? So over here, either there will be an antisense oligo that is introduced. So what this antisense oligo will do, that it will go and sit on some region of the mRNA, the complementary region of the mRNA. And since it is sitting on this region, now what will happen is that it will not be able to bind to the machinery that is required for translation into protein, right? So it will obviously hamper its expression into the protein, and thus expression of the DNA is hampered, right? Similarly, there could be other such cases in which the mRNA expression is hampered, such as the ribozymes could basically cause such an effect, right? Or the risk complex that is basically produced due to the fusion or due to the incoming effect of a small interfering RNA or a micro RNA or an shRNA that is artificially produced and injected, could basically again lead to a complementary binding to the a region of the mRNA such that this region is then degraded, right? So either it will just hamper its ability to form the protein, or it will maybe degrade the mRNA altogether. So thus, this particular expression of the gene is lost. Thus, that is what is known as gene silencing. Okay. So, since it is not a very big slide, I will discuss this also. So, what is the central dogma of biology? It basically means that the genetic flow of information is unidirectional, that is from DNA to RNA to protein, right? Also, one gene codes for one polypeptide. This was something that was originally proposed as central dogma. And who actually originally proposed central dogma is something that we discussed in the previous session. It was actually proposed by Francis Crick, right, in 1957. But now we know that the original thesis that he gave, that was actually not true entirely. Okay, instead what is true is that not just DNA, but even RNAs and proteins can also self-replicate. Apart from this, the occurrence of reverse transcription proves that the flow of information is not unidirectional, right? Even from RNA, DNA could be formed. Viruses can exist without DNA, and hence it is not always the genetic material that propagates the information. Also, there are non-coding RNAs and ribozymes which exhibit properties similar to that of either a gene or a protein, right? So this is our current and updated understanding of the central dogma. Also, when we talk about self-replicating proteins and RNAs, so RNAs, we know there are self-replicating RNAs that are present in the microbes such as viruses, bacteria, etc. But can anybody tell me about self-replicating proteins? Like, what are these proteins? And we will discuss
Prions.
in today's, yeah, so they are prions. So these are basically kind of infectious materials, infectious proteinaceous materials that have been found or discovered, and they can basically cause, they can basically self-replicate. They have this ability to self-replicate. Based on that, there are hypotheses for proteins that have come up, such as the prion model protein aggregation, which does not really have anything to do with the original prion being found, which is an infectious particle. But okay, I will not confuse you further. It is a separate thing to discover. So maybe you can go and read about it.
The prion, how exactly it is self-replicating, what is the mechanism, what is involved. You can give it in the next functional genomics course, not this course.
Okay.
Thank you.
Okay.
Fascinating.
Yeah, sure.
Field.
Okay.
I have a very quick, small question.
Yes.
In shotgun approach, is cloning required, and then how many times one has to clone? In shotgun approach, we fragment the DNA.
In the shotgun, yes. So cloning is required in that, but how many times I am not really sure about. Like what is your question? Can you please frame it again?
In the shotgun approach, once we have fragments of DNA, then we go and then sequence them. Are we supposed to have that DNA multiple copies or not? Whether we have to clone them multiple copies to get the signal or not? That was the question that I had,
Okay.
Or only once fragment of the DNA is there, from there maybe a couple of cycles of cloning of that itself is sufficient. Or we have to do multiple copies. That is number one. Number two was we heard Sanger's method and this next generation sequencing also. We cannot call Sanger method was required for developing the next generation sequencing. Are we using some of the technology from Sanger to next generation? Is that the flow or not?
Yeah, so in the first case, although I'm not sure if that is the only way it could be done, but in most of the workflows that I have actually gone through, cloning is actually a step that is always involved. But I am not absolutely sure if that is the only way it could be done. I think that is done for increasing the efficacy or efficiency of whatever reads we are getting, so that they could be aligned properly. But maybe I will look into it. In the second case, yes, that is the workflow, because when we talk about next-gen sequencing, for example, it could not really have been possible without Sanger sequencing, because in next-gen, we are still lacking, like whenever there is some kind of an analysis that is going on, we are still lacking some, there are some gaps that are still achieved between sequences, because there are a number of sequences that are simultaneously being sequenced, and now we just have to align these sequences. So alignment is also possible, but sometimes there might be minute gaps that might be obtained in between. Now, how these gaps are filled is by basically looking at the human genome sequencing project, which has already been done and which has given an outcome. And since that was actually possible because of this particular Sanger method, I believe that it had to have preceded this
Thank you.
particular
Thank you.
yeah, NGS. Okay. All right. So thank you for the lecture, and thank you for staying throughout this